Welcome back. Now let's talk about the cell, the basic building block of life. Cells are the smallest independent living entities. Cells, smallest living entities. They are small. Some cells are one cell small. Some bacteria, we call them unicellular. If they're one cell, they're unicellular. Others, like humans, have many cells. We call them multicellular. For example, we humans, we have 30 to 35 trillion cells. Now, how much is a trillion? That's a big number. If we want $1 trillion, we're gonna take $100 bills and stack them on top of one another and go all the way, 200 of them make an inch. We go all the way from New York to Chicago. Look at that, like that. And that gives us $1 trillion. That's a very big number. I don't really understand that. And we have 35 or 30 to 35 of those trillions in our body of these cells. So cells themselves, then one, if you look at that organism, is not that big. One of these cells is tiny. It's very small. And we measure that tiny thing in microns. We measure them in microns. Or there's also a micrometer. It's a micrometer. We can write that like that. And what is a micron? A micron is 1/1000th of a millimeter. And a millimeter is like this little thing here. It's tiny. Or it's 1/10000000, 6 zeros, of a meter. A meter is about this much. So that's tiny. That's very small. The smallest one we can see with the eye is the ovum. The one that we reference a lot is the red blood cells. These are red blood cells. So one red blood cell is around 8 microns. An ovum is around 120. And we can see that one with the naked eye. So what we certainly need some serious magnifying glasses to see these little small tiny things. That's for sure. Oh, and then before I forget, the term cytology is the study of cells. All right, good. Let's now talk a little bit about the microscopes, of how we can see these things. Thank God, a long time ago, father and son duo in 1590 figured out how to look at these tiny little things and created the first microscope. We mostly now use light microscopes in the classroom. Light microscopes. The term micro means small, and scope means to look at. You know by now, I like these terms and take them apart a little bit. So there we go, a light microscope. We have a stage, and the stage is where we're going to put an object, a slide, with a specimen on it, and then to magnify it, we have a eyepiece first, and the eyepiece here, where we look through, we can have it either a single thing or a two of them, so we can see three-dimensional that way. And that eyepiece magnifies it, the object 10 times. And then from there we also have an objective. We have multiple objectives that magnify the object further. We have one that does 4, we have one that does 10, one that does 40, we all the way go to 100. to a total magnification. Then if we add the 10 together with those, we can get up to 1000 magnification. Oil is then used between the slide and the lens at the highest power, because at that point the lens and the slide, they touch each other already. Having then those two sets of lenses, another thing that it does, it flips the image over and it turns it right and left. So what is right shows up left, what is up shows up down at that point. Then we have the term magnification. The term magnification indicates the degree of enlargement. So magnification is the degree of how much we enlarge something. Fication. And then in the other term we're going to have here is the term resolving power. And the term resolving power, oops, sorry about that. Let's make that a big one. Resolving power is the ability to distinguish two objects as separate. In light microscopes, we have a light source down in here that brings light and carries it through the specimen up then into the magnification, and then we can see the object. The object has to be translucent, we can get light through, or it has to be very thin that the light can still penetrate. For me and a lot of other students, one of the hardest things is to focus an object. And we have two focusing wheels. And so generally first, we're going to use the coarse focusing wheel, the big one, oops, so there we go, the big one, the coarse focusing wheel. And we do that to orient the object on the stage right, and we do that at the lowest magnification power, just to get situated and more or less get it fine-tuned, which we then do with the fine focus one. So we can fine-tune the focus with that knob, that's the small one, and when that turns, it's not as fast moving, so we can really zone into it. At a higher power, then we just use the small one to fine tune to get the clarity of the image. And not everybody's eye is exactly the same, so when you go from student to student, you do the fine tuning and you use that. That's once it's sharp, we look at something like that. We see all those little dots in there. Those are all the centers of cells. So you see all these little things are all cell centers, a nucleus. Are those. And then if you want to see something bigger or even smaller objects, make them visible, we can use the electron microscopes. You also have the electron microscope. Look at that, I left this in there for the R, microscope. And that's great. We can see things like that. We can make them three-dimensional. What we do instead of light, we use a beam of electrons that move back and forth across the cell's membrane, showing us a three-dimensional surface. See the red dot there? Those are actually Salmonella bacteria. All right, this is sort of how we can see the small little tiny things and the instruments we get from them. So next now, look at the cell themselves and look at all the structures inside a cell.